you've had a whole 24 hours without having to listen about metabolism after having had like 3 lectures on on these things in a 24-hour period. So hopefully you've had a little bit of a break, but also haven't forgotten everything. We were just talking about these sorts of things, right? How does changing one enzyme in a pathway affect itself and how much control it has over metabolism, as well as how much it affects everything else. And hopefully you remember at least the punchlines, and I'm hoping that both from the lectures as well as from being able to futz around with R, a little bit about the intuition. Because of the curvature of these things, you make the world better, but you're not so important anymore. Everyone else now becomes relatively more rate limiting as things go on. So what actually happens when you bump up? So you take one of these enzymes, you bump it up. How does it kind of affect things? And and getting a little bit back, I want to with the last couple slides on this get to why don't we see more effect? I kind of qualitatively hit it, but I want to just kind of hit it one more time. And so you can go ahead and do this if you want to yourself. I'm just going to flip to the next slide where I've already done so. But you can play around with changing things and ask how what else changed. In version 2 of the MCA time dynamics, I've got a couple extra plots that I think are useful, and so we're going to look at those right now. So what happens upon changing an enzyme? So same story as before. We're picking on enzyme B, and this gets doubled between 60 and 40 in terms of time. What sorts of changes do we see? So here's looking at fluxes, but what I've done, and maybe you guys at least remember the order of magnitude, right? The flux is about, what, 0.2, I don't know, 7 or something like that, the steady state flux here, right, on that order. What I've plotted now in this slightly more complex version is the forward flux of each of these in blue, the reverse flux in red, and the net flux in black. And what you can see actually is the net flux is often kind of tiny compared to the total. And so for some of these, the last two, and if you remember, these were the two that had somewhat steeper control curves. They matter, so to say, more than the others. They all matter. This is a world in which every enzyme should be valued. But here, at least, there's the forward, there's the reverse, there's the net kind of thing. But for the top two, you can see the scale. The total reaction is actually quite large. It's like 15 going forward and approximately 15 going backwards, with just a delta between them of 0.27, right? And so, a lot of that enzyme activity is getting wasted. When you bump up the enzyme, what kind of happens there? Look at, here's the enzyme that got changed. You basically just like double the amount of useless work that you're doing. Now you're doing a ton going forward and a ton going backwards, but the net, that black, just went up by whatever it was, like 5%, just a little bit. There's very little kind of proportional impact, and all the others become just like slightly more effective now than before. And so this is one way of seeing it in terms of the kind of the differences between them. That just, in this case, VF and VR both become larger. But we can look at that either as the ratio of forward to reverse fluxes, or I think the most interesting for me is thinking of kind of like the catalytic efficiency of all the damn reactions that you've actually done to substrate or product, the total. So F plus R, the forward plus reverse, how much of it actually moved in the direction of net flux, the F minus the R? And I put it on a log scale so you can even see it, because they're so pathetic. There's nothing magical about the log scale. But for the red and the orange, it's about half. So it's not too bad. Like the net forward is pretty decent. So about half of their reactions are going forward, and the other half is like 1/4 each of ping pong going back and forth that cancel each other out. So still, not as efficient as you'd like it to be, so as to say, but that's just kind of how it goes. But if you look at these guys, like they are horrifically inefficient, so as to say, in like 1%, 2 or 3% of their total activity is actually going in the net direction. And by bumping up that one enzyme, you just made it really inefficient. You've cut its efficiency by half. The blue one dies down and then pops back up. And the sad thing here is, remember, we have 4 enzymes. What's the average control coefficient? 1/4. This is a really positive scenario in terms of how ineffective enzymes would be. If we made a series of ODEs to describe an entire cell, where the average control coefficient is like 1,000th, like almost every single enzyme is going to be in this, not particularly effective zone of things. It's really kind of depressing, so as to say, but like this is how catalysis works. And there are a few checks upon this that make it a little bit better. Like I said, there are some enzyme reactions that are relatively irreversible, things like that. But this is kind of the way that things manage to work out. And so kind of the last way I think to think about these things is we've got the equation for kind of the flow rate going forward and reverse. And you've got the forward Vmax and then its saturation term, and the reverse and its saturation term. And I think it's worth thinking about these each kind of separately. Right? These say something about the maximum capacity for flux. And often is where the target of mutations would be. Why? Because Vmax has both the enzyme level, and very, very, very commonly in lab evolution as well as natural evolution, changes in expression play a huge role, or Kcat, the actual catalytic efficiency of the protein is wrapped into there. The only spot that the quality of the protein lies outside of that is in its KMs, the saturation constants. So then we have these 2 saturation terms, which are largely dependent upon the entire system and not that enzyme alone. All right? And so I think it's useful to think about when you changed one enzyme, how is it that it was ineffective, yet it was able to make other enzymes flow better? And so we can think about, obviously, any enzyme has a range of activities possible. Where V could stretch anywhere between Vmax forward, could be the best it could possibly do going that way, or it could be negative, in terms of Vmax reverse, as fast as it could possibly work backwards. It's going to be somewhere within that space. And as we saw from kind of catalytic efficiencies, it's often kind of near the very middle of that, somewhere not so far from 0 compared to what it could be doing. And so if we think about kind of the original system at the 1x level of EB, if we think about kind of the relative enzyme levels were kind of 1 to 1 to 1 to 1, kind of just at the background levels, right? We knew that the flux, so let me see how do I want to write this. We'll have flux in red. The flux was a relative flux of 1, and we had simply relative enzyme concentration of 1 and the saturation term of 1. So like kind of like if we're just kind of making everything relative, what would it be then at 2x, right? At 2x, let's say the flux jumps, the steady state flux is now something like 1.2. So if there was a 20% increase, but now we have a ratio of enzymes of 1 to 2 to 1 to 1, relatively, compared to what they were before. How did we get there, right? What caused that to happen? And from the point of view of enzyme levels, enzyme B doubled, and enzyme C didn't change at all, right? So what caused those two differences? It's because in terms of the relative saturation, enzyme B is now relatively less saturated than before. The only reason why the flux is higher than it was is because at least like 2 times 0.06 gives you 1.2. It doesn't just give you 1, but it's much less saturated than before. So that's why you don't get kind of the full benefit that you think you'd get from making a change in any given mutation, any given enzyme. How do you make an enzyme that you didn't mutate go faster? It's now better saturated, right? By that exact kind of same proportion. Does that make sense? Right? And so any given enzyme has to kind of, by changing any mutation to an enzyme, has to change all the other kind of saturation terms to rise up, all of them to rise up, as it's also dropping down until you reach a new steady state. And often, I mean, this is actually a pretty rosy scenario where a twofold increase gives you a 20% change. If this was like 1.01, then that's almost basically 0.5. It would have almost been exactly the reciprocal. You may as well just throw almost complete waste at it, and the saturation over here would have just gone just a little bit better on average for the other enzymes. Does that make sense? It's kind of like a heuristic for thinking about these things. And so this is why metabolic systems are inherently quite robust to change. The control over flux is well distributed over lots of enzymes, many of which have relatively little play. So what does this suggest about enzyme saturation, right? So in theory, then from what we just said, like none of these enzymes are fully saturated. If an enzyme was fully saturated, you could do nothing to change its flux. It would be completely kind of unchangeable, right? So what would be the kind of scenario that would get close to an enzyme being fully saturated? What would you have had to do to those enzyme levels for you to say, I changed this enzyme and it's really close to fully saturated? What kind of change might you want to make? Remember what happens when you turn up an enzyme, you turn down an enzyme. What happens to its own substrates versus its own products? What would it take for it to be like really saturated in the forward direction and not really saturated in the other direction? You'd have to turn it way, way down, right? And so if in the middle of a chain, you had a really awful enzyme, either really low expression, or it's from some foreign organism, or it doesn't have very good activity towards this particular substrate, et cetera. If it was really low, it would fit that extreme where, all right, it's got a lot of substrate built up and not very much product because it's so damn slow making product and the enzymes beyond it can use them really quickly. But unless you're in that kind of extreme, enzymes should not be very saturated by theory. What do the data look like? All right? And so to be able to measure metabolites is a total pain in the butt. If you're curious about it, I can describe it over a beverage of either daytime or nighttime variety. However, one can make these sorts of estimates. And here's some nice data from the Rabinowitz lab where they're plotting metabolite concentrations. Note the scale. So this is in actually in molar. Millimolar would be then right there, which is a fairly decent guess. A lot of metabolites are in the millimolar range. Many order of magnitude differences. And then here's Km values, the saturation constants for different enzymes, also over quite a different range. So these can be all over the place. The x equals y line, right? If somehow every single data point was on the x equals y, it would be that every metabolite in the cell is at exactly the Km value of the enzymes, right? And then the one line that's offset from there is 10 times the Km, and the other one is 1/10 the Km. And so basically all of that is unsaturated. So where it's like less than, say, 10 times the Km. So certainly that already describes a lot. But you might very clearly argue, well, doesn't it look like there's a fair number of cases where things are saturated? So shouldn't there at least be some 20 or 30 enzymes that are perfectly saturated? That looks to be the case. However, the math we were doing in the R code was for one metabolite, one substrate going to one product. And unfortunately, a lot of metabolism doesn't just have one thing going to one thing. You get math that looks like this. right? Where you've got one here and one here. So even if, say, NAD is really high, it could be perfectly saturated. If this term's unsaturated and is like 0.1, you're still nowhere near saturation, right? For the enzyme to work at its full pace, it needs both of these things to be around, at or near their high saturation constants. So this is going to cut out a large fraction of those. And the math, as these things get to be more and more complicated enzymes, can get worse and worse, and you can have even more terms involved. So most enzyme substrate pairs are unsaturated and those with one substrate high often have the second substrate low. All right. So the reality does indeed look, from that perspective, like what we might think. But how else might we test MCA experimentally, right? Besides just going into R and writing some ODEs and playing around with things, like, does it work the way that it should? Is this the right realm of things? Are the approximations that go into the way we modeled and the way we think about it reasonable? And there's kind of two different ways of doing this. You can do this in vitro fairly simply. If you have a way of assaying flux through a pathway, like you add some substrate, it goes through a few enzymes, and then it turns something over that makes a color or does something interesting at the end, you can ask, all right, put different levels of enzymes and ask, give it a little bit of time to reach a steady state and see, all right, what level, what rate do you see go through that pathway? And then you can dial in some system, dial in extra levels of one of the enzymes. Or hold the other ones down and dial in extra levels of the next one, or extra levels, et cetera. And so indeed, you can find things like if you do this with glycolysis, that hexokinase, as you dial in more, you actually get quite a good big response. Phosphohexosisomerase, it really doesn't matter. You can just add more and more and more, and it has such a small control coefficient, it looks essentially flat. So that's one way of getting at it. The other way is to do the same thing in vivo. So instead of just adding different enzymes to a test tube, you can regulate things up or down. And so you can do this, well, you can have things be up or down. There's two ways of doing it. One is to use a regulated promoter so that you can add some chemical that induces a system to various titratable points. And you get data that looks something like that. That we'll look at something like that in just a moment. Or you can use discrete mutations. If you have particular mutations that you know have distinct levels of one or more enzymes, you can look at that and see how well it compares. This was some classic work from Tony Dean. He and Dan Dykhuizen and Dan Hartle in the 80s had a series of seminal papers in this area that really asked this question in terms of, are these thoughts that come from metabolic control analysis, does it seem to play on, in reality? And they found it actually, being able to do pretty well explaining their data. This is how to draw a 3D landscape back in 1989. And then, with an analytical approximation from MCA, they have, kind of biochemistry trying to predict fitness, and it actually fits the data pretty well. So we're going to take a look at a particular example of this, where we're going to carry on from where we just were when we were talking about epistasis, really, right? Does this look familiar, I hope? This is where I was asking you, two different changes on enzyme expression, what would happen jointly? And indeed, the expression of the double looked a lot like the two pairs multiplied against each other. It fits the data quite well. That's nice. Molecular biology is not always so kind. In this case, things looked well. Not so well for fitness. Two great mutations, either you just don't get any benefit from the second one, or it even gets worse. And so, what's going on there? Can we learn from this from metabolic control? Right? Why is there antagonism and even sign epistasis? Sign epistasis seems really hard to imagine. Every curve we've seen so far looks like this, like olive green one, where as you dial in more enzyme, it just saturates. It might become flat and boring, but it's monotonic, like it doesn't go down. other than when I was talking about negative control coefficients and branch points and stuff. Let's leave that to the side for now. But generally speaking, they don't go down. Okay, then why would you have, where would you get sign epistasis on a nice, smooth, monotonically increasing curve? That doesn't seem to make much sense. But unlike when you write this in R and you just change a parameter, like you didn't have to pay, right? You didn't pay rupees for each amount of enzyme that you were using in there. Unfortunately, the cell does have to pay. There are many costs to making enzymes. One of them, the straightforward one, is the economic cost of synthesizing more protein, but there's also kind of the opportunity cost of using ribosomal bandwidth. Those are kind of relatively predictable and set a baseline. No protein can be expressed for less than that. But as usual, life can just get worse. And so you could have an enzyme that also catalyzes a toxic reaction, or uses up some cofactor you needed elsewhere, or misfolds and causes the cells to look awful like we did, right? So there's a whole variety of ways that you can have enzyme costs, and in a number of studies, people have found those costs tend to rise approximately linearly, not surprisingly. Or maybe have a little bit of a tail eventually when just like you reach a threshold value and the cell just can't take it anymore. But for a lot of studies, it looks relatively linearly, and I think you can already see where this is going. If you have this increasing linearly, if this has downward curvature, you're going to end up with a peak in the middle, right? Can we understand this? And so we did a model for this. This was former graduate student Nigel Delaney, and it's really a fairly simple kind of approximation of metabolic control. And so we wanted to think about kind of the aspects that have to do with kind of the metabolism of enzymes, and then also kind of the cost of production. And so there's kind of three parts to this model. Let's talk about this part of things first. So this is trying to approximate with one term the metabolic control result, right? Every single metabolic control curve we've drawn, even though like you had a system of three not so pretty looking ODEs, but they generated these nice little hyperbolic looking curves. We can approximate that hyperbolic curve with something that looks just like this. Notice now in this, it's not S over S plus Km, it's E over E plus E half max. So we're saying like from the point of view of the pathway, as we dial the enzyme up, we know that there's it has the same shape, and we can have an E half max, that's the amount of enzyme that would give you half maximum flux through the whole pathway. Make sense? So we can just kind of approximate it as that. Now there's this little, so that would give kind of that nice hyperbolic curve. There's a nice little cheating term here, which I will get to in another slide. We noticed something very unusual about our data. So this is a threshold value of enzymes. We kind of were subtracting off a chunk. And so what was very weird about our data, now, remember every single metabolic control curve we've looked at before, where does it go to 0? Where does this function hit 0? At 0. If you, in principle, if you have one enzyme in the cell, you should have flux. It'll just be really damn slow. And if you had two enzymes, it would probably be exactly twice that much, and then it would taper off. But that's where you should hit 0. In our data, and actually some data that Deepa did when she was in the lab, in a very kind of similar system, we didn't see that. We went like halfway up our range of expression before you got any growth at all, and then very quickly going from like half to like 55% expression, boom, it shot up. That it displayed a threshold, and I'll talk about in a minute where that threshold comes from. Here, in that math, we just wanted to essentially cheat, put in a single term that takes into account this aspect, that there seems to be a threshold amount of enzyme before you can grow at all, which I will discuss in just a minute, and then cost for the two enzyme. Now, does anybody notice something funny about that little algebraic expression? You notice that there's something missing. How many enzymes are represented in costs? 2. There are 2 steps. They are both required. If you delete enzyme 2, they will not grow at all. yet I don't have enzyme 2 in the equation. Why not? It turned out, even though our mutations affected enzyme 2, and actually affected enzyme 2 by a lot, none of them brought enzyme 2 far enough down to actually make the flux go down. So like those control curves that go da-da-da-da-da-da and then shoot down really low, we didn't have any value to fit like where exactly it crashes to 0. And since we didn't have that regime in any of our data, we just ignored it. It's not to say that it doesn't matter, but we're basically just saying it's a flat enough control curve for enzyme 2 that we simplified the math and took it out. Make sense? All right, so just one algebraic expression, but it would give us this kind of brownish curve in between, right? Because we've got it slid over, curving up, and then subtracting off a line, and so it's going to come up to some intermediate optimum and then have a slope coming down. And so that's what we're saying, but it is actually two-dimensional because it would be in both E1 and E2 space. What data do we have? So the nice thing, these are dots. I'm going to show prettier pictures of it later. We have a number of different points in the landscape for which we have fitness values. So the fitness values are all those numbers that are indicated. The ancestors way up there, high level of expression of both. We had inducible promoter, all those gray dots. So we turned from high levels of expression, we turned it down, 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 down. And you can see what happened. The values go kind of up to about 20%, and then down quite a lot, down to about 58%. So we had those values, and then we also had some various single mutants. And what we're doing is we're going to fit these parameters with those single perturbations, and then ask, can we predict the combinations? That's kind of the logic of testing this sort of idea. And ask if we can predict combos. So fairly similar to what we talked about yesterday. All right. Yeah, actually, and that's what it says here, in terms of using some of the points to try to predict the other. And there was a fair amount of data to predict both of those. And you can see kind of the model landscape looking kind of straight down on it, fitness indicated in color. Let's talk first about that threshold, because it may... Yeah, please. Yeah. It's actually Flux. Yeah, I know. Yes. It is a fair point, actually, that there is... Yeah, good call. It's making... Thank you. Even though I know that to be true, I have had it wrong on my slides for a long time. Thank you. That is absolutely the case. We're using a vertical displacement in the y to shift the damn thing over in the x. That's a very nice way of putting it. In principle, we could have subtracted it off of the e sum threshold in both places. But yeah, that is absolutely right. Thank you. Why is there a threshold at all? Right? This is what things should look like. If you're at some given expression level and at some given flux, everything we've been talking about looks exactly like this. Okay? That's in terms of flux. But what about in terms of, say, formaldehyde? It turns out the enzyme involved, I don't have it here, the enzymes in the foreign pathway are the ones immediately downstream of formaldehyde. So if they're too low, what is the substrate that's going to build up? It's not just anything, it's formaldehyde, right? And so what happens is you dial the enzyme to concentrations, right? If we now plot concentrations, we'll start at some point here, is we turn the enzyme up, what happens to its own substrate at steady state? If you have more enzyme catalyzing reaction, will its substrate go up or down? It goes down a bit. So it's going to do that kind of a sag. But as you turn the enzyme down, it's going to go up by a lot. So that's fine in the computer. Your computer doesn't mind. But what if the cells care, right? What if there's some threshold of formaldehyde that, right, you're dead. You know, so that anything up here, you'll be dead. What's that going to mean? It means it's going to cut off a range. It means that there's going to be, once I've brought the enzyme down low enough to right here, you know, this zone would all be lethal due to high concentrations of formaldehyde. Does that make sense? Like from the point of view of catalysis, it's a perfectly fine solution. I could be right here, and I've got a flux, it's just slow. The problem is I have a slow flux in a dead cell because it's caused the intermediate to build up so much. And so it's important, like to grow, cells need to turn food into biomass. That is certainly true. We'll talk more about kind of things from that perspective. But you also have to do so in a way that doesn't kill you. And so like that is a constraint, but then you can have these extra constraints. And this is one type of extra constraint. As far as I know, actually, it's the only metabolic control curve that's been shown to have kind of this type of phenomenological behavior, so that the net effect, and just about out of colors, it was a curve that looks more like this for fitness. You know, something like that. Make sense? All right. So how well does this model do? It's a very simple little algebraic expression, and we already know that the simple model of just treating the cell as a black box doesn't really predict fitness very well at all. You do that, and you ask how well does it predict the doubles that did not go into fitting the parameters. It gets it actually like really well, really, really well. You know, the neat thing about this, and I think it makes metabolism kind of nice compared to a lot of things like protein work, is that you have these really continuous, knowable, canonical shapes to the fitness landscapes. And so here's a view of the fitness landscape, kind of from the side. There would, in principle, be a cliff. So this is levels of the second enzyme, FGHA, and levels of the first enzyme. You do need this enzyme. And so it should actually make, you know, looks almost like Half Dome in Yosemite, for those of you who are familiar with that kind of image. Like there would be a fitness cliff on this side, too, but it rises up and falls back down. There's a threshold because of toxicity. It rises up as you need more and more catalysis to be able to make things flow fast enough, and then by the time that saturates, it tilts back down in both directions due to enzyme cost. Sound good? One thing that's neat about this, now that we've got a picture of this fitness landscape, we can ask something, for example, like, why didn't we ever see any promoter mutations? Like, we got these other weird mutations that were like between the two genes or on the plasmid or things like that, and not kind of the obvious class that is, you know, common in some other experiments to simply see promoter mutations. So I think there's a couple ways of looking at this. If you stare kind of straight down on top of this landscape, we often think of adaptation and fitness landscapes from a hiker's perspective. I'm somewhere, and I'm like a little cloud of hiker, you know, variation hikers, and whichever direction on the landscape is most steep is the most likely to kind of pull the kind of cloud of hiker variance up, and then it would kind of march up to the top of the ridge, and then kind of like along the ridge until it reaches the peak. There's a very kind of local, leftover from Fisher sort of feeling of you know, little tiny changes. And for some phenotypes, they may, you know, when it's highly polygenic and stuff like that, maybe that's fair. And so if that were true, the direction of the locally steepest gradient is kind of that way. If there was something simple, like a one-to-one correlation between the two enzymes, which would kind of be a null expectation for a common operon, that you'd have twice as much of both or half as much of both, things would have changed that way. But it turns out the global optimum is in this direction, turn down one some, turn down the other by a ton. And all of our single mutants managed to hit this space where there were these kind of oddball mutations that in a single step turned them down and adjusted their ratio. Like, it's actually pretty cool. And they, so instead of thinking of adaptation as being, you hiking on a mountain, think of it more of like spraying out, like rockets, like all over the landscape. And those that are successful, like, can just, take off from there. And if your rockets can reach the peak in one step, then like forget everything that's in between, right? Like, at least from a phenotypic point, you can just get there and kind of nail the peak in one step. And this, I think, gives us insight into the interactions. Why did we didn't see kind of the null expectation, which would have been in green, that stacking two good mutations together would have been better than either single. We sometimes saw them just simply kind of flatten out, and other times it got much, much worse. We can look back and see why that was true. And so I pulled out two particular pairs to illustrate this, one from each of the classes, the expression changing class A and the copy number class B. For this first set, A3 and B2, they both drop expression down to kind of modest amounts. They're both beneficial. When combined together, expression is simply multiplicative, and it slides down further. But look at the curvature of the hill. Like, it's gotten kind of to the edge of the cliff, but it hasn't fallen off. The other pair, B5 and A2, this B5 mutation has a fairly similar fitness to like that A3, for example, but it already is kind of right on the cliff edge. You combine B5 with A2 in terms of expression, and this one pulls down by, say, 40%. It pulls this one down by another 40%. But you've gone flying over the cliff, right? This is the intuition for why you certainly don't see as much benefit as you expect, and sometimes they're even net deleterious. It's because there exists this kind of, this cliff sort of function, and this could easily happen also for protein stability. This is exactly what I was talking about. If you're already near the threshold of falling off, you're much more sensitive, even though like you may have just as much activity as some other strain that's here, but the next mutation that pulls you down, when you have kind of a thresholdy type of selection, this kind of thing can happen. All right. So, a few evolutionary implications. Dominance. We heard about dominance, right? This isn't just like the worm stuff. I think this gives us a chance to think about dominance, at least within metabolism. What's your expectation? Metabolic diseases, are they dominant? Are they kind of like right in the middle? Or are they recessive? Recessive, why do you think? They do stay. But is there something you see about this curve that would also give us a hint as to why they'd be recessive? Well, even just in a single individual, I think we can interpret this curve. Like, let's imagine like 1.0 represents big A, two good alleles for an enzyme. All right? That's how much enzyme activity you have. That's how much flux through a pathway. Where would like big A, if little a has no activity be? Right? You like that, for example. Because of the curvature, and this one even has some like decent curvature built in, right? There's just potentially a relatively small amount. And given that most enzymes in metabolism have such shallow control coefficients, like I said, the average is something like 1/1000th. So these are really, really, really flat. So as long as you have just some enzyme, there's often relatively little effect. Two bad enzymes, all right, now you've got a problem, right? And indeed, it's like essentially a true statement to even be this absolute, that all metabolic diseases are recessive. all of those that affect enzymes in this sort of way, unless it's some sort of bizarre dominant negative that it does something wild and wacky that's different. But if it's a loss of function, which are most of them, loss of function alleles, they're going to be recessive. Sound good? Let's say you want to take advantage of that knowledge and make money. And you want to say, well, instead of disease is where we think low activity is a bad thing. Let's imagine you have some pathway you want to stop, right? And you've got some choices of which enzyme to hit in a pathway, where this is wild type. Right? Which of these enzymes should you design a drug for? If this is A and B. Which one would be useful to design a drug for? A, right? For A, if you can inhibit it, you'll see an effect, right? Like it doesn't even make sense to start going down drug discovery with B if it has such a steep curve that I need to inhibit that enzyme 99.9% to get like a 50% reduction in cholesterol synthesis or something, then that's just not going to help. I take statins every morning. They inhibit hydroxymethylglutaryl-CoA reductase. I teach biochemistry and I can say that phrase. And that happens to be the most, that's not the only, but the most rate-limiting step in cholesterol synthesis. So it works. You can actually make this work. You need to target where something that's going to be useful. So even a modest inhibition, you can get a big effect if you target A. As a side note, kind of flip side, which, if we imagine this was adaptation now and the cells need the enzymes, which would be the better candidate for beneficial mutations if you wanted this pathway to happen? If there's selection for flux. So to say, if you want to create a deleterious situation, hit A. But now you want a beneficial situation. Which enzyme do you think is a more likely target for beneficial mutations?
Some would say also A.
Yes, it's also A, right? A matters, and it matters more in both directions. It's the only one that has any steepness left in kind of the good direction, and it's the one that has the most effect in the other direction, right? And so in this case, it's actually the exact same answer. A. That you can imagine some beneficial mutation, and you can see, obviously, if it affected B, not much happened. A, much better, right? Kind of the game we were playing before with the code. Lastly, and I think this is interesting in light of what we were hearing about earlier with worms, thinking about kind of variation in populations, et cetera. I think, at least with regard to metabolism, this can explain kind of a pretty interesting finding. So Hubby and Lewontin, 1966, perhaps some of you in this room even might know what that paper has to do with anything. This was actually one of the very first views at a kind of a molecular level at variation within populations. And so these were different flies, and these were individuals. And what in the world am I looking at here? You can tell that's a gel. Hopefully you can tell that's a gel. It turns out this is actually an activity gel. So it's a native gel. You haven't unfolded the proteins. They can run in their native form, and its activity happens to be of alkaline phosphatase. But what was interesting, all these flies were essentially wild type, perfectly fine in terms of fitness. But some of them had like 100-fold more enzyme activity than others for a key gene. And okay, we understand that there's variation in a population. There's beak size, there's tail length, things like that. But compare the variance to the mean for most traits, right? height in this room. What's the coefficient? Like there might be, what, 10% variation, coefficient of variation or something in the room. Like it could be some range, but not 100-fold, right? Like this is crazy level of variation. Why doesn't selection see this? Normally, we're much more kind of constrained for such traits. Not so for metabolism. Why not? These damn curves are so flat. It doesn't matter, right? The reason why you have such huge variation, so this indicated wide molecular variation in populations. This was long before sequencing was possible. And what these control curves say is basically, if you're up here, you're essentially neutral within reason. Now, what I just told you about the work that we had done with methylobacterium, you'd say, well, doesn't it slope down strongly? And it could. It depends upon how costly the enzyme is. And so there will slowly be selection against like massively high levels. But actually a lot of enzymes, biosynthetic enzymes in particular, are expressed really pretty low levels. And so even if the cost per enzyme was kind of high, there's so little of it in the cell, it doesn't, it's not going to be kind of so powerful. And really for a lot of them, the control coefficients are so flat, you can get by with relatively very little. And so you can have really quite wide variation in the population because of this. That behaves effectively neutrally. Because of this high standing variation. In the few cases where you see actually tight variation around some enzyme level, you should look into it because it means that there's something kind of funky going on. And there are a few cool examples of that kind of stuff. And so, yes, please. Mm-hmm.
So does this signal that there are less number of traits which are related to each other, because had they been related to each other, then there's some sort of balancing selection and you would expect less sort of heterogeneity as compared opposite to this case.
So I do think this is an important point. Remember back to when we asked about, what if you changed all enzymes in a pathway? Remember, if we doubled all of them, what happens to flux? It went up twofold. So if you have, say, a regulatory mutation that makes an entire pathway, every enzyme in that pathway, go down, they can't kind of compensate for each other. So those kind of mutations will certainly break this kind of rule, right? And so if you have whole sets of things that are changing, and indeed, a lot of the global regulators of metabolism turn up all the enzymes of the TCA cycle, turn up down all the enzymes for histidine biosynthesis, things like that, kind of in a coordinate fashion. And there's yet other interesting reasons why controlling things in a coordinate fashion makes sense, since a lot of phenotypic heterogeneity arises through transcriptional bursts. So at least you bursted the entire pathway high or entire pathway low, which, as we just learned, keeps all the metabolites just like they were. And so, like, that was a complicated little extra tidbit, but for those who care. So I think you absolutely could have changes that would have larger effects, but they're generally going to have to be coordinated changes and not just the single individual ones. Or, I mean, obviously, a larger individual change. Yeah. So I've tried to throw down kind of some of the bullet points of things. Don't worry, when I give this for my class, there's a test. For you guys, there won't be a test. But these are at least some of the kind of the take-home points. Are we ready to leave metabolic control behind? Perhaps some of you want to leave it behind forever. But hopefully, you can see that this metabolism, and we're going to look at metabolism from a totally different light and a totally different kind of math. I think it's the kind of physiology that offers at least some hope to relate some levels of phenotypes up to other levels of phenotypes. Now notice I didn't say genotypes. It's not like I can tell you turning that G to a C in that codon of that gene is going to move an enzyme activity by some amount, right? That's a protein biophysics and enzyme catalysis question. I think it'd be really cool if we someday get to the point where we could actually predict that, and if we could predict what effect the changes would have upon traits, just let it rip. Then you could actually predict kind of all the way up soup to nuts. But at least I think this gives us kind of a chance to try to move from one level up in kind of a synthetic way. All right, so now, I know this isn't tutorial day, but too bad, I've got a challenge for you. We're going to talk about a different kind of a metabolic model. This is called flux balance analysis. That'll be written out on another slide. But it's basically asking this kind of question. Imagine the cell could use 12, I'll go ahead and read it out. Use 12 molecules per time. And so that first, you could use a number smaller if you want, but let's assign this first flux to 12. What should be the pattern of fluxes that would give you the most possible cell material? I want as much of those 12 to end up down in this bucket. Now, at first it seems trivial. Well, just send all 12 to that bucket. That'd be too easy. So here, this is your test. See that metabolite Y? Think of Y as being like a cellular currency, something like NADPH, ATP, something like that. You need to make Y somewhere so you can use it. This pathway makes Y over here, right? So you need, clearly you can't do 12 to 0. You can't do 0 to 12, otherwise you won't make any cell mass either. So what's the best solution? Six and six? So we've heard an 8 to 4, we've heard a 6 to 6. Oh, and let me explain one last thing that might be confusing. Imagine biomass requires 2 molecules of D and 1 molecule of E to make like a little brick of biomass. So they have to be in that ratio, because that will certainly temper the answers too. Come on, you guys are like quantitative, amazing geniuses. This should be easy, right? Yeah. What did you say, 9 to 3? Yeah. 9 to 3. All right, let's play that one through. So then what? What would you do with the split after B? 6 to 3, 6 to 3. So kind of if we add up the 6 plus the 3, we kind of got 9 of it, so as to say, into biomass. Okay. 3, and then should we send it through C to G or C to F? Here? All right, well, let's try it for a second. This is fun. This way he won't get too excited about being right. So how many Y molecules were made? You made 3 Y here, and that's it. How many Y molecules do we need? 6 Y. We're short. But if we swap those, right, now, does that make sense? Right, like everything, so first of all, like these solutions, they're balanced. This is at steady state. In and out of every one of those nodes is equal. Otherwise, the math would not be fair. And including kind of the Y, I didn't draw like arrows over because it would just get too messy. And indeed, this is actually the global optimum. We'll do the 8 to 4 in a minute. So this happens to be the global optimum, the best you can do. This is the viewpoint of flux balance analysis. It is totally separate set of questions, separate math, separate way of looking at the world from the metabolic control. It's asking exactly like this, like just step back to what's possible for the entire cell, figure out what kind of steady state is possible, and amongst those, which would be the best. All right, so that's how this is going to work. Right, and so metabolism doesn't look like that. It looks more like this. Again, don't worry, it won't be on the test. So to spread out and do flux balance analysis, it is not 1,000 different ordinary differential equations with the associated 5,000 to 8,000 parameters and things like that, thankfully. That would have its own advantages, but certainly has its own disadvantages. So when we think about metabolic pathways, what does flux balance end up ignoring? It says, well, let's not explicitly consider enzyme activities. We just know that they exist. So as long as I know that they can happen, I don't care how much, or what their Vmax is, or what their Kms are, or anything. As long as I know that it can happen. Okay, that certainly simplifies the world. Also, there are no metabolite concentrations. What's left? What's the one thing I haven't crossed off? The fluxes. So this only considers the flows. Only. Does that make sense? Like, all we're concerned about here are the flow rates, right? It'd be like telling you, something about, like, some sewer system. Just tell me how much water is flowing through everything. I don't want to know how big the pipes are. I don't care. I don't care how deep the water is in any of those systems. Boring details. I don't care about Bernoulli's equations. Just tell me how much flows in each direction. That's how flux balance works. And in a nutshell, it works something like this. You have food. You have biomass. Food goes into metabolism and then goes into biomass or into waste products. And there are 3 steps to what goes on. First, there are constraints upon use. I'm very glad that our cafeteria food has not looked like this picture here. The main constraint upon, it uses the known metabolic reactions. That's certainly a constraint. You aren't allowed to invent anything. And you cap at least one flux for transport into the cell. And usually this would be carbon, but it can be whatever. So we might say, kind of like in the thing there, we might say, all right, the cells can use up to 10 millimolar of glucose per unit time. Great. Given that, what's the best possible solution? Because otherwise you could just say infinity of everything. Like, you need to stop the world of possibilities somewhere. And so you can strain more than one, but usually only one of those borders will end up holding the cell back. Then you've got the network topology. So really, I guess I didn't need to write that into the constraints. So it's the known network and, really importantly, the biomass composition. Right? In my earlier examples, I needed to tell you that you need 2 Ds and 1 E to make biomass. If I didn't know what biomass was made from, like, what should be the goal of the cell? What am I optimizing? What is this factory supposed to be making? And then there's a third step, and this is really where this becomes an evolutionary optimality model, is that you optimize, you find the optimal solution for biomass accumulation within the feasible space of steady states. Okay? So this is really different than what we were doing before, right? Before, with metabolic control, you have an actual system, usually smaller, with actual details, and you have a time-dependent, you've got kinetics, as we'll see. This is going to be purely just steady-state solutions, just numbers, and you've got, like, concentrations and fluxes, and you can change parameters, et cetera. None of that is going to happen here in the same sort of way. So what is the math of flux balance? I'll do some of this. I'm not going to do a substantial amount of this, but here's kind of a toy example. Here's a little network of A, B, and C where, like before, these are metabolites, the V's are fluxes. So you can, of course, write this in this crowd. It's lovely. I don't have to explain what that means. From my biochemistry classes, Lord knows I need to explain what those little dAs over dTs are, right? So you write these as ODEs. And then what? We can simplify the notation, express this as a matrix times a vector. so that you have a vector of concentration change per time, the dA dt, dB dt, dC dt, equaling this S thing in the middle. We call that the stoichiometric matrix times the flux vector, right? And like I said, I don't think I need to explain what vector times matrix multiplication is. This is just really lovely, and the first time ever that I've explained this, and I don't have to say what that is, right? So this is the flux vector, just in terms of the terminology, and we call this the stoichiometric matrix. And just to be clear, right, what are the rows and the columns of the stoichiometric matrix? What, if I were to pick out a row, who's that? What's that row represent? Effect upon B. That's metabolite B. What would a column be? So like, let's pick a column. Exactly, right? So each column is a reaction. Each row are the metabolites. So if you see A1, it means that, so like in V2, it creates, there's a +1 in the C row and a -1 in the A row. V2, over there, takes A to C, right? That's what it is. As you can imagine, so it doesn't look too bad here. This is big, right? This is like several hundred by several hundred when you do this for the entire genome. How sparse is this matrix? Another question I never ask my biochemistry students. Really, really sparse, right? Most reactions are using like 2, 3, 4 things, and every other entry is a 0, and vice versa, right? So they're very much, sparse matrices, but contain essentially all of the topological information for the entire network. Mainly 1s and -1s. Occasionally you'll get a 2 in there or something else, but that's mainly what it is. All right? Yeah, this is nice. Okay, so then you solve, right? And any set of algebraic equations is always solvable, right? What's one reason why that might not be solvable, uniquely solvable? If it's underdetermined. And unfortunately, these are underdetermined. You do not get an answer. And instead, you get a space, right? And this is a convex polyhedral cone that is sliced off by the uptake constraint, all right? Again, the only audience who would ever use those words when trying to explain this. They just do it like this. But there's going to be some constraint of uptake. They don't even draw it like a convex. I've got a picture later of it, of uptake. But everything within here, those are all feasible steady states. Okay, what have we done? So, I mean, it's kind of useful. The steady states say these are all, if our network is true, conditioned upon that statement, this is every way the cell could possibly be living. Everything from every flux equaling 0, the cell's dead, out to everything else, right? Okay, but like, I want an answer, right? What do you think, within all of that, what answer do you think is right? And for that, you've got an optimality criterion. What was our optimality criterion for the first question? Most biomass. And so that you do the same thing. You've got one set in your stoichiometric matrix, which is the biomass reaction. And that is not just integers. It'll be like 4,000 carbons is usually how it's stated. There'll be, 33.3 acetyl-CoAs and this many tetrahydrofolates and this many pyruvates and this many this and this many that, et cetera. That's kind of all the exact ratio of recipe that you throw it in a pot and you've got E. coli biomass, right? And so you need to know what that is. Once you know what that is, this becomes linear optimization for that. And as you guys probably know, the solutions will always be upon an edge or even an intersection of edges, et cetera. So sometimes you'll get a single point. Usually not. If you've got degeneracy in the system, you'll get like a line or a whatever. But that's the basic idea. let's make it a brighter, more exciting color. So you pick the optimum as kind of the guess of what the system is doing. Now here's the question though. Why? It's fun to pick the optimum. Why should it be the optimum? What would be the rationale for the world should be the best that the world could possibly be? That this factory works absolutely perfect. Selection. So this is an example, I mean, there are many types of optimality models. This is an example of an optimality model where you frame, phenotypes to fitness in some mathematical way, and you ask, well, if there was selection upon these properties, what would be the best thing? Or selection upon those properties. And people have used this from trying to predict what should the shape of a tuna be? Based upon hydrodynamics, what would be the most efficient way to swim forever? Or what should be this shape? What should be these sorts of things. If a fungus is trying to launch spores, what shape should they be if they're this size? All this sort of stuff. Here we're asking, well, how should the metabolism work to possibly give you the most biomass? So this is ultimately it's an evolutionary optimality model. So it assumes past selection for optimality. All right? Well, so often they're trivial. So imagine you have two enzymes that do the exact same reaction. Then you have a linear combination of the two fluxes. Fine, I guess not a very exciting difference. You can occasionally have two entire pathways that are stoichiometrically about the same. You could go this way and it only uses 188 DH. You could go that way. They go through different intermediates, but yeah. So often you get a space of nearly equal solutions, and they're usually for these relatively trivial reasons. It cannot be multi-peaked in reasons that I can't prove, but maybe someone in this room can. But it will either be a space that's all equal or one point, but it won't be like this totally different solution or that. This is a linear optimization. All right. So what would a suboptimal example then have looked like? So we can begin to see kind of how would we test whether that first one could be true. So suboptimal, let's, I'll pick on Lucy since she said so many other good questions here, so I hope you don't mind. Let's take the 8 and 4. All right, let's play this one through. What's the best we could do? If 8 goes to B, it's not as nice anymore, not integers. 5.3 to 2.7 about, which means we need 5. Let's make this blue so we can see it. 5.3y. And those will come down through here, and 8 total kind of chunks went that direction. What happens this way? We got, this will give us 4. That's kind of great, because before we were only sending 3 there. So that first step is actually giving us more. But we only need 5.3. So how much do we send each of the other two? Right? You can't just go send all 4 here. It won't be balanced. You're making too much y. It's not a steady state. Right? And so unfortunately, you've got to be a little bit wasteful. And so this is going to have to be 1.3, yeah, and 2.7, I think that's right. So that we get an extra 1.3. Everything's reasonable in terms of our y values. And so now we've got 2.7 going out and 1.3 going out. Remember from before, we didn't have any F going out. We didn't spit that out. Now we do. So biologically, let's say an efficient metabolism like respiration, yeast might spit out only CO2. And in a more inefficient sort of metabolism, maybe they spit out something that's only partially utilized like ethanol. And so that would represent kind of a balance of the two. Now, I like the fact that they're inefficient, but this kind of difference is what gets reflected in what would be predicted to be spat out. All right, you guys are smart. Constraint of 20 there. This is obviously nowhere close to as complex as these. This is why you let the computer do all of the things that it does, right? But you can imagine this gets a lot worse. I'll go ahead and throw this down rather than make you do it. Well, I feel a little disappointed in myself for saying that. So the optimum is to split 16 to 4, 4, 4, 4. Because this kind of has this interesting dissimilation is now happening in kind of 2 spots. If we want to think of the kind of production of cofactors is kind of like dissimilatory metabolism. Burning off food so you can make energy and reducing power so that you can keep the rest of your food as biomass. Each of those kind of represent that. And let's see, so then this would be 0, 0. We make, let's see, 4y there. We need 20y there. So we need 24y. And it's a good thing that this set of numbers sets up to have 12, 12, 12, 0, 0, because you can now make 12y here, 12y there. Oh my gosh, isn't that amazing, right? So this happens to be the optimal solution. You've used dissimilation and a perfect balance with assimilation that kind of optimizes things out. Like before, you can imagine things having been different. Here's one already written out. Here's 20 and 6. So interestingly, taking in more of some, like the second substrate, you can think of it as being like nitrogen. So maybe that's glucose and ammonia or something like that. And so let's say you want to go ahead and bring in more of the second, if the cell did this, let's take the word want out of it. If it was now 6 and a molecule of C and B join together to make J, it means only 14 go that way, 6 there. And ironically, then the cell can only actually assimilate 2. It drops its biomass in half by having kind of misbalanced the use of the 2 initial substrates. And suddenly, we see this being spat out. We see this being spat out. We still have that. But it's dropped everything differently, right? So we can see that the pattern has changed. What you spat out has changed. How much you grow has changed. Right? And so if we were to look at this as what like this bigger version is, here is our stoichiometric matrix. I'm sorry, well, you can't, the color doesn't come up very well anyways. If you're red, green, colorblind, you're not missing too much. But I've got all the 1s and the -1s for everything going on. And so the flux vector, where by the time it multiplies against the stoichiometric matrix, it means that when you add up all those products, they all have to be 0, right, for it to be steady state, except for the boundary fluxes. Those are just coming in or just going out. You don't have to play, they're not part of the Sudoku game of like making sure everything is perfectly balanced. They better not be 0, otherwise you're not doing anything, right? And so there will be a net in and net out of various things. So they're outside of this kind of everything being in steady states. Just like the metabolic control where we kind of defined S and P are outside of our system, we're just gonna concentrate on the stoichiometry of what's inside. The same thing is true here. You've got the outside world that you're just drawing from, and you throw into, and you kind of just ignore those parts in terms of the steady state of the inside. Yeah. So these, yeah, I already wrote do not need a sum to 0. So what could we do? What do we do to test this kind of model? There's a couple tests. One, a trivial one to some extent is you could test knockouts, mutations, so where you just completely get rid of a gene, and you can ask about things like viability. And those have gone reasonably well in the past when people have applied this to real data. But really the more subtle and the more direct test, and ironically the one that hadn't been done so much, is you can test, well, do you actually see the flux vector that you expect to? If this is a flux vector that would be optimal, and this is a flux of the suboptimal, like which do the data look like? Did they look like the optimal or some other set of values? Yeah. And then you could kind of plot, for example, what you predicted versus what you observed. If you had an optimal set of fluxes, it'll be right on the line, and if not, there will be some deviation from there. You're not quite, what you predicted. So, do you think this works? How well do you think this works? You know, there's a couple challenges here, I would say. Quite a few. One is we may, of course, not know the topology correctly, or we may not be completely correct about the optimality. What are other, I would say there's at least two other issues. What was the basis for thinking that it should be optimal in the first place? So that's the criterion, right? And so I'd say one issue is that the right criterion? So the maximum biomass, and it's biomass per what, actually? What were we normalizing to? I was saying you got to use like 20 of some substrate. So it's biomass per substrate. If this was a car, that would be in units of, whatever, miles per gallon or kilometers per liter or whatever, right? It's yield, actually. It's efficiency. So we'll talk about, like, is that a good idea? But there's even a kind of simpler one. What?
Enzymes?
Do you know what?
Like for the other costs.
Oh, the cost, yeah, no, absolutely true. There could be, that's a good point, that you could have like some costs, et cetera, that would constrain you from being able to get there. Like why? So it assumes that there's been past selection and that selection could reach that optimum, right? And it assumes that there somehow does exist a set of kinetic parameters that would make this happen and that you could reach and not be stuck by costs. So that you have had selection, it's possible to get there, it's not somehow constrained, and this is even the right criterion. That sounds now a little bit more problematic, right? So is this the right criterion? Is biomass per substrate? Has there actually been sufficient selection on that substrate in the past to reach the optimum? And is the optimum even possible? And this could be either constraints, like you were saying, in terms of cost, or just the kinetic parameters, that there doesn't even exist a set of kinetic parameters that could make the system behave that way. All right, so how should we test this? Can I just take a strain from nature? You can, people have. Test wild type, and as we'll see actually, wild type E. coli, where this model was developed, it actually performs pretty well. With lots of other systems, it's okay, not as great. But if you just take some natural strain, right, I isolate some soil bug on some compound, and I'm going to test it on that compound. Fine, I know it can grow on it, but am I sure that it's actually going to be optimal for that? Like, I don't know its past history. What is a model system for we know the longest number of generations of its past history that you've heard of many times before in this last 10 days, or 8 days, or whatever it is, right? The Linsky lines, the E. coli lines, they've been on glucose since perhaps before some of you were born. Right? And so if it's ever going to be true, you kind of hope it would work in this case, right? And here I've got a drawing that is little, here's our little convex polyhedron cone or whatever it is exactly, right? Like we want to ask if that's the optimum, and somewhere in here we've got the ancestor. Does it, maybe each of the different lines might take a different path, but do they evolve to the optimum? Right? That seems to be a reasonable question. And what about this issue of the criterion? So, as mentioned, the way that we're maximizing things in flux balance is biomass per substrate. It's an optimum efficiency. It's like the Prius kind of view of the world, where everything behind is just green hills. What feature have you seen several of us show as kind of the primary component of fitness, at least in the batch culture, where there's plenty of resource around? Speed, which would be like biomass per time. Anybody ever heard of a rate yield trade-off, right? Like, who's to expect these things to be positively correlated? They might even be negatively correlated, right? If we've had 60,000 generations of evolution to be a Porsche, and yet we think it's going to have the gas mileage of a Prius, like that seems a little hard to understand whether that's actually a reasonable guess for such things. So a former postdoc of mine, Will Harcombe, a good friend of Deepa's, we tested this. This was going to be just, you're new in lab, why don't you just learn how to grow some things and do some of this while you're starting your other projects, and of course it's many years later. But eventually I think that's what I actually feel pretty happy about. So we looked at the long-term lines. We know that their rate for all of them have increased. They grow faster. The rate of glucose uptake is certainly a lot faster. But no one had actually looked to see like what proportion of carbon, like there's a proportion of carbon kept higher or lower, and what pathways are they using for doing that. And so we used a technique here that if anyone wants to know, kind of like I said about metabolites, if you're curious about how could you use C13 labels to follow metabolism through and get back at all these flux ratios simply from steady state data, I'm happy to tell you about that another time. But he was able to measure kind of each of these key branch points. How much of glucose goes down typical glycolysis, goes over into what's called the Intner-Duderoff pathway or the pentose phosphate pathway, which way does it get into the TCA cycle, what splits are going on. And so he assayed this in the ancestor, everything in this heat map's relative to that. Higher fluxes in red, lower fluxes in blue. And you can see here are the evolved lines. If they simply live their life exactly the same relative fluxes, just do everything faster, they'd all still be white. They wouldn't have changed at all. They'd just be faster versions of white because these are flux ratios. So they have actually like changed what it is they're doing and how they run their cell besides simply being faster. So there were significant changes. Those are the little dots within there. Are they did they all do the same thing? Well, there's certainly some things that are more common. A lot of these turn this down, or some of these things are a little bit certainly higher than chance, but are they all the same? Certainly not all the same. They're different from each other. And then lastly, how does it compare to flux balance predictions? This would have been the set of changes to reach the optimum. You can see, visually, right? Like that don't look much like that, right? Like certainly it's, if anything, for a number of these fluxes, it seems to be in the wrong direction, right? So let's take a look at that. So we can look at this in a couple of ways. We can look at just the overall yield. Given the set of fluxes that are happening, how close to optimum are they running? And for this bar chart, the black is the ancestor, and then the evolved lines, these are 10 of the different 50,000 generation lines. So this has been a fair amount of time. I'll talk about the 50,000 generation lines actually again when I get to trade-offs on Friday. So how well is the ancestor running? The ancestor is actually at 98% of the optimal value. Like I'd have to say, not bad. Good on you, E. coli. That's pretty good. The evolved strains, did they totally turn into Porsches during the selection and have really low efficiency? No, it actually didn't go down very much. It is statistically significant, but it's certainly not substantial, right? So surprisingly, we didn't actually see a rate yield trade-off of any substantial amount. But as said, the fluxes are not quite what you'd expect. So the ancestral fluxes compared to optimality are in the triangles, and like I'd have to say, that's pretty damn good. That such a big model with so many assumptions does that well. Like I'd say that's pretty good. The interesting thing though is, of course, all the evolved lines are actually like a little bit less good. Again, they're not horrible. They're not doing things completely different. But if anything, they've actually moved away from optimality, away from what would have given them the optimal biomass. So it's kind of glass half full, half empty, right? It actually does a pretty damn good job of predicting the ancestor, but the direction of evolution was surprisingly in the wrong way. I'm almost done here. Is that the only possibility? No. And so there had been prior work from the lab that kind of developed flux balance, ironically, where they evolved strains and measured how much they improved, and they made flux balance predictions and then compared those to like transcriptional data, but didn't actually compare them. And they had flux data in another paper, and they never compared their actual flux data to their flux balance predictions, which seems a little bit odd, I would say. And let me complete that triangle at this point. And so they evolved E. coli on lactate, right? This is what makes yogurt sour. And E. coli, it don't like lactate. They grow slow. They grow to much lower yield. They're at 80% optimality, and they grow pretty slow. They improved a fair amount in terms of rate over a relatively small period of time. And interestingly, in this case, the evolved lines, at least on average, actually did improve their yield at the same time. So they actually did get better in both rate and yield at the same time. So if we look there at the fluxes compared to the predictions, the ancestral are somewhat more spread off this line. We did statistical tests not shown here compared to the evolved lines. And so they actually did move. So in this case, the ancestor wasn't very well predicted. Flux balance would not have been a great way of guessing E. coli on lactate, but it actually was a good guess for where evolution would move. And we looked at a few other data sets, and there was this interesting kind of, again, half full, half empty way of looking at the world. The closer the ancestor was to optimality on that substrate, it would evolve away from optimality. The further away it was from optimality, it would actually evolve toward. And so, certainly, these flux balance predictions are, I would say, in the realm of what the cells actually do. They should not be taken sensu stricto, but I would say it's not bad. I think given the massive number of assumptions involved, the fact that you can make these sorts of predictions at the level of yield, at the level of the fluxes involved in these strains, is actually not too bad. And I'll come back, I'll use flux balance analysis a little bit when I talk about trade-offs on Friday too. Yeah? Oh, no, this was, I'm sorry, this was from Bernard Paulson. Yeah. And like I said, they compared flux balance predictions to transcriptional data in one paper, and they had their fluxes, so we just used their flux data from yet a different paper, but they didn't compare the fluxes to the predictions in any of their papers. And which I think is kind of unfortunate, because I actually think it's a reasonably optimistic-ish, it doesn't say FBA is perfect and awesome, but I actually think it's fairly optimistic there, that at least we can get a decent guess of how things would go purely from first principles. And again, should you care to look, I've kind of tried to make some bullet points in terms of kind of what the main take-home message is of how this works and the ways that this can be used. And we will talk about it a little bit with trade-offs and a little bit, if we get to that point, in terms of kind of coexistence and using this to predict equilibrium frequencies in a multi-species community, and ask, spatially structured even multi-species community, and ask whether it can do us any good at all. All right, I am actually caught up. So thank you very much. Let's call this a day.